if you find that you've got a problem with your related substances and you only had it at a certain time point, then you might want to dig back into your raw data to see, well, is it the data or is it the way that it's being manipulated that's causing the problem? And always look, if there's anything, always be suspicious of something that's happened when you've changed. So for example, when you go from a manual capture to electronic capture, well, it's unlikely that that's a trend. It's more likely that it's the change in the way that you're capturing the information. Similarly with nitrosamine, that's being identified as a significant issue at the moment. The actual data, to me, isn't likely to be different. It's the way that we're looking at it that's changed. When you do a validation of a test method, so 10 years ago, you were probably going to, your limit of detection likely was higher. So you can't say that that validation that you did 10 years ago is invalid. It's just not up to current standards. And what people fail to do is consider that things move on. And they say, oh yeah, we validated it when we first did the production of the product in the first time. But then somebody comes back and says, well, we've got this new related substance. Well, where's that come from? Well, we changed our HPLC to UPLC. And because we've got increased a lower limit of detection, we can now see something that we couldn't see before. That doesn't mean your original validation wasn't valid at the time, but what it means is now you can see things that you couldn't. And then what people fail to then do